Ooh, it's recording. Okay, so we're going to start with the ribosome. And questions we want to ask about all of the structures would be something like, right? So it says eukaryotes are larger, more complex ribosomes than what? The prokaryotes, right? Pretty much everything in eukaryotes is more complicated than prokaryotes, right? Prokaryotes a little more simple, right? We're here before us. What does the ribosome do? It chills. It does what? Protein synthesis, right? Which just means makes proteins. What are proteins made out of? Yeah, amino acids. Oh. It's disappearing. Okay, amino acids, right? Linked together by peptide bonds. All right. Where does this happen? The cytoplasm. And on the rough ER. We'll get to that. Are these a membrane-bound organelle? Nope. Right. So they are made of ribosomal RNA and some proteins. And they're a big, giant complex. The small subunit and the large subunit. Right? Woo! Oh, what the? Oh, what's happening? Okay. All right, and then we'll get back to ribosomes a little bit more when we get to the ER. Oh, here's the ER. The rough endoplasmic reticulum. These are membrane bound. So pretty much the ribosome is the only one we're going to talk about that's not membrane bound, right? All the other organelles or all the other cell structures are membrane bound, right? What do we mean by membrane bound? Phospholipid bilayer. Semi-permeable, same stuff that we learned about plasma membrane in the organelle membranes. It is continuous with the nuclear envelope. Since I just posted that and you didn't get to watch that, the nuclear envelope is just a membrane, the nuclear membrane. They call it an envelope, I don't know why, because it actually has two phospholipid bilayers. What? It has a double membrane. So they call it an envelope instead of a regular membrane, trying to make it a little bit different from the rest of the organelles that are only single membrane. So the ER has one single membrane. It's still a bilayer, right? Remember the phospholipids, tails together, heads outside. They call it the rough endoplasmic reticulum because when they took pictures of it under electron microscopy, right, there was all these little balls stuck to it. Right? It's not smooth. It's all rough. And then they realize, oh, those little balls, that rough stuff, were actually ribosomes. So ribosomes are sitting on the membrane of the rough endoplasmic reticulum. And what does it do? And if ribosomes are sitting on it, it must have something to do with protein synthesis. That's the ribosome's job, right? To make proteins through translation. And so, yeah, that's its job, right? So the proteins that are made in the rough endoplasmic reticulum, the guys that go in there, are destined to go to some other membrane in the cell. And we'll talk about that in a second. And where does it do it? Right? So you can see the little picture of the cell and the purple stuff. It's continuous with the nuclear envelope, the nuclear membrane. And when you watch that little video I just did, they have a nice cartoon showing that. So right at the junction around the nucleus where the ER starts, it would be really hard to tell what's ER and what's part of that envelope, right? And we don't really care. We just know that that's pretty much all together, okay? And then also, remember, we do have some free ribosomes in the cytoplasm also, and so the proteins that are made in there are cytoplasmic. They float around in there. They're not going to be in a membrane. Okay. Ooh. Okay, so what does secretion mean? Ooh, what do we know? Do we know what secretion means? Do we have an upload? What? do we have, I don't know. Do we have an upload set up for today in Canvas? Can somebody look for me? I thought I set them up, but now I'm not sure. Yes? Okay, whatever the first one is in the module, define secretion. Or take a wild guess. Based on context cues. So the proteins that are made on the rough ER are destined, are going, to be either inserted in the plasma membrane as a membrane protein, or localized in the membrane organelle, right? They might go to a lysosome. Ooh, it's going to go to the Golgi. Talk about the Golgi. So anything that goes into the rough ER is going to end up in a membrane or it's going to be secreted. Ew, the secretions. Okay, here's a clue. Snails have mucousy secretions. Yeah.
Is this for iPad upload 12?
Whatever one's the first one in the module. I don't remember what name it was. Yeah, just give me a definition of secretion. Right? Because if we don't know the definitions of these stupid words, then you're not going to be able to answer questions. Right? What's the destination? Location, where they're going. Synthesized, being made. Right? Insertion. Localization. The locus, where it is. Okay, are we in? Did we text? Did we put a text in there? Okay, I'm going to tell you the answer. Does anybody want to shout it out? How about another hint? How about, we'll call it cell spewing. What? Cell barfing? Ew. That doesn't look right. Anything that gets, ooh, how about cell spitting? Anything that gets spit outside the cell, yeah.
Is it like, it's kind of like, how do I put it? It's kind of like it's setting the cells out to like do a certain function?
Yeah, so it's sending something out of the cell. And in this case, our example is a protein, right? We make it in the ER, and then it's eventually going to get sent out. Yeah. Can non-cells secrete things like? Oh, is it secreting exclusive to cells? Yeah, pretty much. I can't think of an example, right? Like when your pancreas secretes insulin, right? It goes out of the organ, but it did come from inside a cell at some point, right? And then sent to the little lumen to like, or bile in the liver and sort of. So it all would start within a cell. Viruses can't secrete anything. They're not cells. I don't know. Yeah, you just look that up. You sneak it up. Yeah, so when we want to send something outside a cell, so like a proteinaceous, you could think of it like a hormone, a signaling molecule, something we want to send outside the cell. Antibody molecules, our white blood cells want to fight bacteria, you have strep throat, they need to make antibodies and send them out into the bloodstream, not just stuck on the cell. Some will stick into the cell, help them drive around and find stuff, but others are secreted, put out into the bloodstream. They would have started being created on a ribosome on the rough ER. Ooh, how fun. Where is it? Over here, right? That's our ER looks like that. Okay. Oh my goodness. The endomembrane system. Ooh. So we're going to talk about this guy and how it works. And so it's rough ER, which we'll talk about. Stuff goes in a vesicle. Ooh, a budding vesicle. Wow, my handwriting is terrible. To the Golgi. To another vesicle. To a membrane. Oh. So endo just means in or inside. Membrane is a membrane. And the signal hypothesis is that in order for, how do we know which polypeptides, amino acid monomers, how do we know which proteins need to be on a ribosome on the ER or a free-floating ribosome? There has to be some way to know. If all proteins that are going to go to a membrane or go outside the cell need to be made into the ER on a ribosome, how do we know which ones need to do that and which ones just float around in the cytoplasm? So it's it could be a hormone. It's usually something signals to tell you, but in this case, the actual signal is on the protein that's being made, right? Remember polypeptide, that's just protein. Oh, fuck. Sorry, I forgot my pencil again. Right, made up of amino acids. And so the address tag, what they're calling a signal sequence, ooh, that was for just here, it reminds us of the NLS. We'll remember that for after you watch that other one. It's part of the protein. It is a sequence of amino acids, a specific sequence of amino acids. I don't know what they are. I wish they would just tell us so I could give you an example, or I could have just looked it up and then I didn't. That helps guide that ribosome to go to the ER, stick on it, and make that protein into it. Right? So something else out there, some sort of hormone-y protein guy has to help him get there. Right? But it's something on that protein. So all proteins that go to a membrane have that same little signal. We'll call it the go to the ER signal. And so here's their little picture of that. So we'll if I can go to, I can't touch it, ER signal. That's what we're going to call it. The go to the ER signal. So they're showing us, here's our ribosome guy, and he's floating around in the cytoplasm. Looks like he's kind of close to the ER, right? Here's, double membrane, right? See, look, zoom in. Oh, shit. Shoot. I can't zoom. What is happening? Maybe I have to go here. Oh, look. Lipid bilayer up there, right? Tails in. Ooh, can you see some bent tails? What does that mean? Unsaturated fatty acids, right? And then another membrane, right? So 2 bilayers. Oh, sorry, just had to bring that up. Little reminder. Ooh, okay, go away. There we go. So that guy's floating around, happy as a little clam. Here's the RNA in the ribosome, and it's pooping out a protein, right? 1 amino acid at a time. And as it's getting more and more, that sequence, if that sequence of amino acids exists, the signal sequence, if that's part of that, right, coded for by the RNA, coming out, we have this, little peptide receptor guy, right? A recognition particle, that's another protein that says, hey, I'm binding to that sequence, and I also can bind to a little pore, a channel in the ER. So that kind of brings this guy, ribosome, to the ER, and now the ribosome poops the protein out into the inside of the ER, the lumen, right? The inside of that tube. If this guy did not have a signal sequence, just a regular old protein, this ribosome would stay in the cytoplasm, and this protein would be made out here in the cytoplasm. It would not go in the lumen. It has to have that signal sequence. Ooh! So what is the signal sequence? Give me a text upload for the next one. All right, that signal sequence is made out of what? I just told you. I told you what it is. It's part of the... It's our favorite monomer. Well, it's our favorite monomer. Oh, my goodness. All right, so whatever the next one is are there two? Is there another upload? Okay, you can just put in the text box, right? The signal sequence is a sequence of dun, 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 part of the It's not part of the RNA sequence, right? That's not what gets recognized. The RNA codes for it.
What's the upload called?
Because I just made a copy of it. And I didn't change the name. So yeah, whatever upload copy, copy number 1, first copy. See how I am? Yeah, question.
Is it methionine the protein that signals for translation?
Is methionine the amino acid that signals translation? Yes. Methionine is the first amino acid put on all proteins. Yeah. We'll get there. But exactly. Okay. We want the answer, right? We know the answer. Right? It's the order of amino acids, a specific order of amino acids. That's recognized by this signal recognition particle, some protein, similar to a hormone, right? Grabs onto the actual protein, right? It's the actual protein being made. So it starts translation in the cytoplasm, not attached to the ER, right? So all of them do that. So if you're any old mRNA, right? You're in the cytoplasm, a ribosome grabs you. Oh, the small subunit grabs you first, and then the large subunit comes in there, and you start doing translation, and you start making amino acids, the first of which is methionine, yes. And then, bunch more. If you make a sequence that is a signal sequence, I'm going to have to look up what it is. So let's just say it's a whole bunch of leucines. I don't know, I'm making that up, right? Leucine. If you have 10 leucines in a row, we'll pretend like that's the signal sequence. So any protein that's coded for in the RNA to make that is going to get grabbed by that particle. That's going to grab onto the channel, the pore. That would be considered a receptor because it's receiving something, and then it's pooping the protein into the ER. Any random mRNA on a ribosome in the cytoplasm that starts doing translation, right? First amino acid is methionine, and let's just say it's a whole bunch of methionines. There's no leucines, there's no signal, that stays in the cytoplasm. So only the guys with that signal, a bunch of leucines in a row, gets to go into the ER. Yeah.
Doesn't a signal sequence also affect like proteins, like where the proteins go?
Where the proteins go later, yes. Right. So well, there's all kinds of zip codes and ideas. So one of which is proteins that end up going into the nucleus, have their own sequence to get them in. Stuff getting out of the nucleus has to have a specific sequence also. Exactly. So yeah, that the sequence of amino acids of that protein determines its shape, right? We talked about primary, secondary, tertiary, blah, blah, blah. But it can also determine where it goes in the cell. So many things. Okay. The Golgi. I love the Golgi. Okay. So the Golgi is a bunch of single bilipid membrane. Yeah. Single phospholipid membrane sacs, right? So this is their little picture, and then this is the electron micrograph of those guys. And that's called cisternae. Who cares? It's a membranous sac, right? Each one of these is one long sac that's capped on the ends. So that's what makes it look different than the ER, right? It's not continuous like our big mess was. Our Golgi is in these little stacked up cisternae, right? And there's always some sort of vesicles, little round guys that bud off of the Golgi. That's the whole point. And it gets the products. What are these? The proteins. From the rough ER, right? So it's in the rough ER, those proteins, and they actually bud out surrounded by ER membrane. That becomes a vesicle. And then that will fuse with the Golgi, and then all kinds of fun stuff happens in there. And then those proteins will bud off the other end of the Golgi into another vesicle and go somewhere else. To another membrane-bound organelle, to the plasma membrane, to the nucleus. And then just some definitions. The cis face is the one that is facing the rough ER. So that's where the proteins would go into the Golgi, and the trans face is the one where they would come out of the Golgi. Well, let's see just what do they do in there. What are they doing? Why do they have to do that? Because they have to do a little, the secretory pathway, right? Getting them out of the cell or to another membrane. So here's our little picture, right? Here's our ribosome with our RNA, and he's pooping the protein into the ER, right? It'll literally bud off. So it makes a little bud, and then it squeezes off and makes, right, a little phospholipid bilayer guy with our protein inside, right? So it just pinches it, and then because of the hydrophilic interactions, the tails are going to want to stick together, and the heads are going to stay on the outside, and it's going to close off, right, into a little vesicle. So just like when we talked about phospholipids, last week, couple weeks ago, right, and you throw them in a bunch of water, they'll spontaneously make a bilayer or spontaneously, put their tails together. Same thing. So all we have to do is squeeze that a little bit right there, and then those guys suck together and bud off. Woo! Okay, I don't know. We'll see what happens. Sorry, bad things happened. Okay, so this guy buds off, and then same with budding off. All it has to do is come close to this membrane, and they'll fuse together, right? So this guy opens up a little bit and is attracted to that guy, sort of shoves his way in and opens up, spitting out the protein into this guy. Same thing happens here. Buds off, squeezes in. Buds off, squeezes in. Buds off, squeezes in. Buds off, and then it can go to the plasma membrane, fuse with the plasma membrane, and spit that guy out. It could also fuse with the plasma membrane. If this were a membrane protein, it would have been stuck in this membrane. And when it got to here, it would have stayed stuck in the membrane. And it would have been one of those proteins that sticks out of the cell that doesn't go anybody. Integrated just means they're all working together. So this can put them out of the cell, right? Completely secreted. Or it can put them to any organelle and shove it inside that organelle. Or put it to the plasma membrane and have it stuck in the membrane. If it was stuck in the membrane of this vesicle, it'll stay stuck in the membrane of the plasma membrane, or whatever organelle it goes to. So anybody who had, right, had to have that signal sequence to start in the protein, and it goes to, because it goes to the ER, means it is destined to go through the Golgi, go to another membrane, or out of the cell. Stuck in a membrane, in another organelle, or outside the cell. That's called the endomembrane system, all the membranes inside the cell. And then, why do they have to do this? Why can't we just go right from the rough ER to the Golgi? Because obviously we're budding off the rough ER. Oh, crap. Because we do stuff to it. But first, I forgot. We're going to talk about a little experiment because we're supposed to be talking about how do we know all of this stuff. All of these were done by experiments, right? We can't see this happening in a cell. Or can we? Oh. Only if we do an experiment. Only if we label the proteins so we can follow them and see where they're going. And labeling just means attaching something. What in the good old days, we used to attach something radioactive. Right? Because when radioactivity degrades and is radioactive, it gives off light. And then you can use a piece of film. I know we don't use film anymore because everything's digital. And light hits the film and turns black. And so you can follow stuff around by taking films at different times. Now, we try never to use radioactivity anymore. We use some sort of fluorescent dye, which fluoresces, usually under UV light, sometimes spontaneously, GFP. Right, so we can follow stuff in the cell or label things and see where proteins are or where they're going with fluorescence, which is not dangerous, unlike radioactivity, which is not so good for you. But these are old, because this was one of the first types of experiments. And the pulse chase experiment, which was always, which is described, I think, still even in our text and was always explained to me. You're never supposed to define something using that word, right? You can't say something is interesting because it's interesting, right? Then nobody knows what the word interesting is. You can't say pulse chase experiment is you pulse something and then you chase it. What the hell are they talking about? So the pulse is usually something fast, right? So if you're in the gym and you're doing sit-ups, or you're doing crunches, and then they're like, pulse, you have to go really fast. Like, it's a really fast. Oh, pulse, pulse, pulse. Right? So it's just a fast, we stick a little bit of radioactivity on a little bit of fluid, and we throw it on a cell, and we wait 10 seconds. And then we wash it all out. So it's like a little snapshot in time, what things are ready to go somewhere, get radioactive, everything else doesn't. The chase is just washing it out with a whole lot of nothing, not radioactive. Right? If somebody would have just told me that, I would have not been confused what the hell a pulse chase was for 20 years. Oh my God. Okay. So we're just going to give it a little fluorescence, label some proteins, and then we're going to wash it all away so it can't just keep labeling. You know, if you leave the fluorescence in the fluid that you're got these cells in a dish, it's just going to keep going in there, and then you're not going to be able to set a time frame. So we label them. Where do these proteins go? Oh, I don't know. And so we see them, most of them now are in a vesicle, right? These are all so dark because they're full of radioactivity. We can see individuals here at 7 minutes, right? So we pulsed, we labeled with the dye or the fluorescence, and then we chased, we washed it all away, and then we waited 7 minutes and we looked at it, and the proteins are in these vesicles. We're like, ooh, that's exciting. And then 80 minutes, over an hour later, we see where did these vesicles go? They went towards the cell membranes. These are a bunch of cells together. I can sort of see the membranes, right? And this is a cross-section including a duct, which is just a tube, right, that carries stuff away. So a bile duct in your liver, right, the secreted bile stuff, right, goes, it's so gross. It goes right into the gallbladder to be stored, and then it gets into your stomach when you eat fatty stuff, right? So the duct is just the tube, and so they're saying, ooh, all these vesicles went right near the surfaces, and they're starting to spew proteins into that duct. So anything that's in a vesicle is destined to go towards the membrane, fuse with the membrane, and go outside the cell. That's how they did, they looked at, started in the ER, see if you can find them there, and then do that same thing. Did they go to the Golgi blood? So that's how they followed them from rough ER to vesicle to Golgi, through Golgi layers to vesicle to wherever they go. Same type of experiments. So that's how we could see, quote unquote, what's happening. And then, okay. All of these membranes are lipid bilayers, phospholipid. I'll make a PP, phospholipid bilayers, right? And so the whole point, right, here's our little Golgi guys, is that we put sugars on them and other stuff onto the proteins. So in this case, they're showing us, N-acetylglucosamine, some mannose, that guy, and a bunch of glucoses, right? And that is usually membrane proteins have sugar molecules on them, and that's it's embedded in the membrane, and that is usually can act as a receptor to get signals like hormone signals, and it's also useful for telling your cells who they are. So my best example of a protein that's glycosylated, or a sugar sticking off on your membrane, is on your blood. Your blood type is based on what sugars are sticking out on a membrane protein. Okay, so there's type A sugar, which I don't know what it is. I should look. I don't know. I don't care. Type B sugar. If you're type O, you have no sugar of that type. And if your blood type is AB, you have both A sugars and B sugar proteins. Okay, and they're just membrane proteins that have sugars on the end, and that's how we determine our blood types. And those became your blood type because they're highly immunogenic. Meaning if your body sees one of the sugars that you don't have, you're type O, you don't have either sugar, and you're accidentally given a transfusion of type A blood, you will die. Exactly. Right? Because you will have such an inflammatory immune reaction to that it'll create a huge inflammatory response, a cytokine frenzy, right? And you will essentially go into anaphylaxis and die. Okay? Yeah, crazy. So if you're type A, you can only take A blood or O blood, right? Because O means nothing. You don't have a sugar. If you're type B, you can take B blood or O. And if you're type AB, you can take any old blood. AB, A, B, and O. If you're type O, you can only take O blood. Right? And that's all about a glycosylated protein. Okay, but I digress. Oh my God. Okay, so, this was somebody said. He said in the front about how we get places, right? Not only do we have to have that signal sequence to get into the ER to go to a membrane, right? We also have other tags, like you said, that help sort proteins into different vesicles to go special places, right? Are we going to this is the lysosome, right? Are we going to go outside the cell? Are we going to go to the plasma membrane? Are we going to go to the mitochondria? Right, where are we going to go? Where are we going to go? And some of that is by what sugars are on the surfaces. Some have a zip code tag in the amino acids along with their signal sequence. They all have a way to get where they need to go, and they all have some sort of receptor to help them once they get there. Receptor is usually just a protein that recognizes the sugar or the tag. It does an interaction, and it allows them to come inside. So in this case, right, these guys are going to pinch off and become that vesicle, and then it's going to fuse with the lysosome, right? This guy, these guys are going to return to the ER. How exciting. Okay. Does that make sense? Right, so they have to be told where they're going to go, right? It's very complicated in there. You can't just randomly make some proteins and hope they get to the right place, right? Our cells are too big for random diffusion or random, the protein happened to bump into the right place. If we think about prokaryotes, they don't have membrane-bound organelles, right? They don't have a lot of extras in there, and they're teeny, 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 tiny compared to our cell, 1,000 times smaller, some ridiculous thing, right? They showed that little picture. So they are small enough that when they make proteins, if they have a signal and some proteins in there to help them get somewhere, they don't have far to go. They don't need an endomembrane system to send proteins to their plasma membrane, right? They're tiny, they're small. The reason we think all this craziness had to evolve is because as the cells got bigger and bigger, random diffusion, make a bunch of proteins, they're not going to go to the right place on their own. The cell got too big. It's not going to just randomly bump, I bumped into the side. I'm supposed to be here, right? Or I'm doing whatever I need to do. So we had to evolve some complicated system of sorting stuff. So people use it like your zip code at the post office, right? Every little town has a different zip code. That's one of the first way that they sort the mail at the post office is by zip codes. Then they use your actual street address, right? So same idea. You have to have some zip codes, you have to have street addresses, and you get more and more specific. So our big zip code that just means endomembrane is that stupid signal sequence that gets you in the ER. Once you're in that ER, you know you're going to get to the membranes. Then other stuff is going to be the actual address. Am I going to the lysosome? I don't know, right? Am I going to the mitochondria? I don't know. Or the nucleus. Ooh, what's my address? I don't know. Okay, sorry, that was terrible. That was terrible. Ooh, okay, enough of the rough ER. There's also smooth ER, and this is where we make our fatty acids and our phospholipids. Woohoo! Right? So those guys are not proteins. I know, I'm killing you. Rough ER, all about proteins. Smooth ER, no, no, no. Fatty acids, what's a fatty acid? The C's, C, 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 C. Ooh. Lots of H's, right? Remember, oh my goodness. Does not like my not pencil. Ah! Ooh, I'm shouting. That's okay, you guys are sleeping. Wake up, hello, good morning. Okay, so same idea, right? Endomembrane system, but his job is to make fatty acids and phospholipids. Is it membrane bound? Yes. Single membrane, one phospholipid bilayer all around. Does its budding and all that stuff, but as far as I know, which I could be wrong, does not go through the Golgi. That's just for the proteins and glycosylation, right? But it buds off, and there's probably signals and all kinds of proteins and stuff like that. And so cell bio 380 is going to have to cover that. We're just going with smooth ER, not proteins, fatty acids, and phospholipids are put together, and a breakdown of hydrophobic toxins. I don't know. I don't even know what that would be. Okay, what else? What else? Peroxisomes. Again, single membrane bound organelle. Oxidation reactions occur. Redox reactions. Ooh. Okay, that's all we care about knowing. Again, cell bio will give you more detail. We don't want to know more detail, right? We want to be as simple as we can on some of this stuff. Oxidation reduction. That's hard. It's hard. Chemistry. Ooh, lysosomes. We love lysosomes. Lysosomes, again, single membrane bound storage or waste processing. Think of it like the cell stomach, small intestine, digest stuff. So it's very acidic. It is there to break stuff down. So you can break apart nucleotides, you can break apart amino acids, you can break apart proteins, you could break apart bacteria, right? Ooh, if we eat those guys and trying to kill them, right? We put them in a lysosome and fry those guys. And lysosomes are in both plants and animals, but plants have a special type of lysosome called a vacuole. Remember that guy when we talked about him in plants versus animals? And so the vacuole stores water. Right? Animal cells don't need that because we're not a plant. Right? Plants have to store lots of water that also helps them hold their shape. So if you see a leaf that's wilted, that means the vacuoles are dehydrated. They're not full of water. When the vacuoles are all full of water, then the leaf and the stem would be standing up nice and tall, have that turgor pressure, right? Because they don't have a skeleton. We do, right? We have a skeleton. All our stuff is just hanging on our skeleton, so we don't have to have turgor pressure to keep from wilting. Okay, so we both have lysosomes that do this kind of stuff, that do digestive enzymes and waste, and then plants also have the vacuole, which is a type of lysosome. But they also have the regular ones too. Some people confuse that. Some books too. Okay. Oh, how do we get into the lysosome? You can phagocytose, which is engulfing like an entire cell or particle, like a, well, that doesn't look like a bacteria. That looks, oh, back. Oh, come on. Mommy. Okay, I'm not going to be able to write bacteria, apparently. Okay, you know what I'm saying. That's a bacteria. That looks actually like a little algae single-cell guy, but whatever. And then if one of your organelles gets messed up, like your mitochondria. I don't know why, too much oxidation, too many free radicals, right? We can degrade that organelle by autophagy, which just means self-eating, right? You can think of phagocytosis as a cell. Cyto is the cell. So eating a whole cell or particle. And then receptor-mediated endocytosis is pretty much how we bring in any kind of big molecules. So a sugar, if we're not sending it through one of the transports, and sometimes hormones, if you want to bring them inside a big protein complex. It's when they say receptor mediated, there's proteins on the membrane that interact with whatever you're trying to bring in. They bind together, and then this guy squeezes closed, right? Makes this vesicle here, giving up drawing, this vesicle here, and then if we want to chew that up, we want to digest it and use its parts, we would fuse with a vesicle from the Golgi with the digestive enzymes. We'd then call this a late endosome, early endosomes, and then it fully becomes a lysosome when he's mature, right? So it got the digestive enzyme stuff from the Golgi, right, to become a lysosome. And so that's where we'd put this damaged organelle would eventually go into the lysosome. If we want to destroy, break down whatever we phagocytose, it goes into the lysosome, right? So it's a trash compactor, right? The breakdown where we put waste, and then we can, or the recycling center, because then we can use all the parts from whatever we're breaking down, right? We might need that. Okay. Oh my gosh. Oh, that's nothing. Okay. Oh, we're gonna die. Mitochondria. Why do we care? Because it's the powerhouse of the cell. Sorry, it's the energy plant. I hate when they say the powerhouse of the cell. But who knows what a powerhouse? It's a power plant, right? It's what generates most of our ATP, almost all of our ATP, cellular respiration, or fermentation, right? Glycolysis happens. It has a double lipid bilayer, double phospholipid bilayer, right? Two membranes, the outer membrane, and then the inner membrane is the guy that's got all the little invaginations, right? The cytoplasm of the mitochondria is called the matrix, right? Because they don't want to call it cytoplasm, because cytoplasm's the liquid stuff inside a cell. So the liquidy stuff inside a mitochondria is called the matrix. These little invaginations of membrane, right? What's this? Where's this? Oh, rough here! Our cristae, and then they're just trying to show you that out here would be outer, and then in here would be an inner membrane. And the other thing that I just recently realized was that all of our pictures of cells, right? Ooh, what's this, a picture? What kind of cell is this? What is that guy? That's the vacuole. Woo! Ooh, over here, what's that guy? That's a chloroplast. So here's our mitochondria. There's always like a picture of 2 or 3 mitochondria per cell. And so that's really, that's always what I kind of thought. There's, 5 mitochondria in each. There's thousands of mitochondria in each cell. Like, that blew my head off. How have I lived this long? Thinking there's 5 mitochondria per cell. Ooh, if you're a muscle cell, maybe you have 10. No, there's thousands of these suckers in our cells. So, of course, you can't draw thousands in our stupid little picture. Same with the Golgi, same with the chloroplasts, right? And they're a lot smaller in comparison to everything else, but, right, you have, like, our cartoons are just to help us understand. But that, like, blew my head off. I was like, thousands? I'm picturing 2. Okay. And we care because we would be dead without mitochondria because of their role in cellular respiration, glycolysis, fermentation, all that stuff, which we will talk about ad nauseum later in class, in the semester, and that it's one of the double membranes, right? Double lipid bilayer. What else had a double lipid bilayer? The nuclear envelope, right? Everybody else, I think, right? Everybody else we talked about either has a single membrane or the ribosome has no membrane, right? It's not a membrane-bound organelle. So far, mitochondria and the nuclear membrane, are the only ones that are double. Everybody else was single. And that these guys, the inner membranes, is pretty much where all the action happens, right? And you want lots of surface area. That's why there's so many inside, because that's where the electron transport chain happens. Woo! Okay, we'll get to that. And then the chloroplasts, right? These guys are in anybody who does photosynthesis, so eukaryotic. So we would think plants and algae have chloroplasts. Plants and algae also have mitochondria, right? All cells have mitochondria. I know lots of times in the past when we say, oh, plants have chloroplasts, animals have mitochondria, they're similar type structures, blah, blah, blah. Everybody has mitochondria, only plants and algae or eukaryotes that do photosynthesis have chloroplasts. Okay, so don't get sucked into that question because mitochondria is everywhere, thousands of them. Okay, so the stroma, oh, is that this? Oh, that's a fat guy. So the stroma is the cytoplasm of the chloroplast. What was that called in the mitochondria? Matrix. Woo! Okay. The thylakoid is the little inner membrane. It's sort of similar to the cristae of the mitochondria, right? This guy, has a double membrane. The outer membrane is the outer membrane. The inner membranes are all of these stupid thylakoids. If you stack them up, we just call that a granum. If you look at a whole bunch of these stacks, it's grana. That's just the plural, right? So these are really nice compare and contrast questions. Similarities. The stroma is to the chloroplast as the blank is to the mitochondria, the matrix, right? Similar structures. Thylakoids are like the cristae in the mitochondria, that kind of stuff. Photosynthesis, convert light energy into chemical energy. Photosynthesis. Light energy, what kind of chemical energy? Sugar. Oh my God. And we'll talk about that too. Okay, keep going. Ooh! Here's our skeleton. What about the cytoskeleton? Stupid. Okay, the cytoskeleton is made out of proteins. Actin filaments are proteins. Microtubules are proteins. Centrosomes are proteins. Nuclear laminas, proteins. Ooh, there's the cell nucleus. Intermediate filaments are proteins. They give the cell shape. So both plants and animal cells have cytoskeleton. It just is a little more important in animal cells because we don't have a cell wall to also give us shape. We don't have a vacuole to swell up with water also to give us shape. All we have is a cytoskeleton and the fact that all of our cells are in tissues on a skeleton helping us organize. So it holds organelles in place and it can allow vesicles to travel along them. So that stupid endomembrane system, those vesicles sort of travel along on the filaments of the cytoskeleton. So they're not just willy-nilly. Like, again, I also always kind of thought, oh, the cytoplasm, it's a big bag with a bunch of organelles and there's a lot of water in there and it's very juicy and floppy. It's very, very dense. Like this, showing this few, it's the cytoskeleton, there's so much of it. right? It's super dense with proteins. And so the vesicles sort of climb along in between, and that helps guide them. Holds organelles in place. They're not just flopping around, right? Otherwise, right, if we put our arm out, all our organelles in our cells would fall down towards gravity, right, or return this way or that way. Right, gravity would affect them. They're held in place by the cytoskeleton. So it's not, the cell isn't just, phospholipid bilayer bag full of stuff. Right, it's very, very well organized with tons and tons of these fibers, proteins that hold everything in place. And so this is sort of a better picture of it, even though the filaments, intermediate, microtubule, much more, but they have to be able to show you what's going on. Shows us that, ooh, rough ER, right? Oh, he's so cute. Ooh, look at the mitochondrion. Oh, look at how cute he is. Okay, so, ooh, phospholipid bilayers, right? So this cartoon's a little bit better showing how everything's sort of held together, but it's actually much thicker. What are those people doing out there? Oh my gosh, we're almost done. We've almost made it. Ooh, these are just pretty pictures so you can get an idea of how thick the cytoskeleton is. So if you're just doing the actin filaments, look at how dense that is in the cell. This would be like the membrane of the cell. Right, it's super dense, right? If we're just looking at the intermediate filaments, right, this would be one cell. And cells aren't round, right? Most cells are not round. Blood cells are round, that's about it. Everybody else that's in a tissue is like flat and hanging on. And then microtubules again, right? This would be a cell there, like that. So if you put all those together, like you wouldn't even be able to see through that. It's so dense with all those fibers, filaments, proteins, cytoskeleton. And then what? Cell wall. Who has a cell wall? Yeah, of the eucs, fungi, algae, plants. And then remember, bacteria also have a cell wall, but it's not the same. Right? It's a different cell wall. Bacterial cell wall is made out of what? Peptidoglycan. Eukaryotic cell walls are made out of what biomolecule? Oh, come on, people. Carbs. Which carb? Think fiber. Cellulose. And these are two cells. So this is a cell wall. This is a cell wall. This is the plasma membrane from one cell, the plasma membrane from the other cell. And so in a plant, when the cells are right next to each other, the two cell walls are what are connected, right? But they still have a plasma membrane. That's another thing that people can get tricked into. Plants have cell walls and animals have plasma membranes. No, everybody has a plasma membrane. It's just plants have an additional structure, the cell wall. Oh my gosh, we're almost done. Ooh. Okay, last upload, you're going to list all the organelles with a double lipid bilayer that we talked about, and then list all the organelles that only have a single lipid bilayer. So you could type that into the text box, or you can write it out, take a picture, and upload. And I think that's it. I'm going to look really quick. So we'll do this one first, but I'm going to stop recording because it's enough already. What's next? Oh, and then for our own fun, list all the organelles that only plants and not animals have, and all the organelles that only animals but not plants have.